le tsebe mamelang mme re lo utlwile le team mme hona jwale le tla bona program tsa rona di se le khutshwaneanyana hobane le bontshitse go data e batla e phaphame ka ditjeo empa le rona re batla hore le pase dihlahlobo tsa rona tsena tseo le tlo tobana le tsona mme shebellang mananeo a rona le tle le bona go a sala makgutshwanyana gore a se ka ja data e ngata empa bohlokwa ba taba ko gore dulang tswelang pele ntse le subscriber tswelang pele ntse le lahlela maikutlo a lona re tla dula re ntse re le arabela ka yona tsela yona ena shebella he.
Hello guys, here we meet again. We are still on movement in out and out of cells. So here we are going to do a little bit workout on how cells behave when they are placed in a solution of low water potential. So here I have got an animal cell and a plant cell. Both of them, they are placed in sugar solution. So this means inside the sugar solution there is low water potential. You know, as much as you know that the plant and animal cell, they both have the cell contents. So those are the mineral salts that are found both in a plant cell, and an animal cell. So this is what is going to happen now. This sugar solution, it has low water potential than the contents of an animal cell. So which means that the water potential inside the animal cell or inside the cell of an animal cell, the water potential is higher than the water inside this sugar solution. So this is what is going to happen now when an animal cell is placed in this solution. What is going to happen is osmosis is now going to take place, and then water inside the animal cell is going to move out of the animal cell and go inside the sugar solution, because water potential in the animal cell is much higher than the water potential inside the sugar solution, which means this sugar solution is highly concentrated of sugar. Therefore, the water potential in here is much lesser than the water potential inside an animal cell. So if now an animal cell is going to lose water to the solution of sugar solution, this is what is going to happen. An animal cell is going to lose the water, and when it loses the water, this is what is going to happen. As it loses the water, now an animal cell is now going to shrink. By shrinking, it means now it has lost water to the sugar solution. In other words, they say this shrinking of an animal cell is called a grenation. Now, because an animal cell has lost water, it has now grenated, which means that this water inside an animal cell, it has lost it. So now that is why even the shape of this animal cell is going to change. The reason why it's going to change the shape like this is because we know that an animal cell has got no cell wall. So hence why the whole shape of the plant cell has been changed. So now let's look at what is going to happen to a plant cell when it's placed in a low water potential. The same thing applies here. The sugar solution here has low water potential than the cell, the contents of a plant cell. So this is what is going to happen as well. Here, an animal cell is also a plant cell is also going to lose water to the solution of a sugar solution. So when a plant cell it loses water, what is going to happen is plasmolysis is going to take place. When a plant cell loses water to this solution, plasmolysis is going to take place, which means it is going to lose water. But unlike the plant cell, the animal cell, an animal cell, it has got a cell wall. Therefore, even if it loses water here, the plant cell is not going to shrink because it has got a cellulose cell wall, which means that cellulose is stronger. Therefore, the cell is not going to shrink. But what is going to happen is, immediately when it loses the water to the sugar solution, we are going to see the cytoplasm is going to move away from the cell wall, which means now the cytoplasm is the one that is going to shrink, moving away from the cell wall. So which means if it loses the water, the cytoplasm is the one that is going to shrink. And also the size of the vacuole now is also going to be altered, because now the contents inside the vacuole, they have now moved out into the water. So now, what is going to happen here, this change of losing of water of the plant cell, when it loses water to the sugar solution, now this is what is happening, is called a plasmolysis. So this means now the plant cell has plasmolysed, but then the shape is not going to change. Remember, even if osmosis have taken place here, the plant cell has lost the water, but then the shape is not going to be altered because of the cell wall. But the plant cell, the animal cell, however, it has granated, which means it has lost its shape. It's because now it doesn't have the cell wall. Thank you very much. So now, let us go on to see the behavior of these cells when they are placed in a solution of high water potential. Okay, here we go again now. But then right now what we are going to do is to show how these cells, a plant and animal cell, they are now going to behave in a solution of high water potential. So now, here we have got pure water, pure water here. So this means the concentration of water potential in pure water now is higher than the concentration of water inside the cell contents, which means that both animal and plant cell, they have got what we call the cell, which contains the mineral salt of those cells. So now, osmosis is going to take place here. And what happens here is, water is going to move from a region of high water concentration to the region of low water concentration or low water potential. So this is what is going to happen now. The pure water now is going to move inside an animal cell. Water is now going to move inside an animal cell. And what is going to happen is, more and more water is going to move inside the animal cell up until the cell is going to expand, and when it expands, at the end of the day, the cell is going to burst. It is going to burst because it has got no cell wall. Therefore, much water is going to move in up until this cell now is going to burst, which means now it couldn't hold on because of this water that keeps coming in, because of the pure water potential which the cell has been placed in. So now, if an animal cell is going to burst because it has got no cell wall, let us see what will happen to a plant cell when it's in a solution of pure water that has high water potential, of course. So now, water is going to move inside the plant cell by osmosis. So now, what happens when this water it moves inside? It means now the vacuole now is going to receive so much water. And when the vacuole gets water from this high water potential pure water, it means now the size of the vacuole now. The size of the vacuole now is going to increase. And when the size of the vacuole increase, is going to push the cytoplasm. So when the vacuole expands, it pushes the cytoplasm. So when the vacuole pushes the cytoplasm is going to push the cell wall. So now, this means plant cell has now taken so much water. And then when it has taken so much water, it becomes turgid. This stage of turgidity is whereby now a plant cell has absorbed more water by osmosis from pure water potential. And then the vacuole, when it receives so much water, is now going to push the cytoplasm. And the cytoplasm is going to push on the cell wall. But because of the cell wall that has covered this plant cell, a plant cell is not going to burst like an animal cell here. But what is going to happen is that the cytoplasm is going to exert a pressure on the cell wall. So now, this pressure that is going to be exerted by the cell wall due to movement of water inside the cell is called Tuga pressure, which means turgidity can lead to Tuga pressure. The pressure exerted by the cell wall, as water moves in, the cytoplasm expands, and then the vacuole pushes the cytoplasm, and the cytoplasm pushes the cell wall. So here we can see that the cytoplasm now, the shape is now bigger, but then the cell is not going to be, it's not going to burst like the plant cell because it is now covered with a cellulose cell wall. Thank you.